My name is Rebecca Kamerer, and I have been working with Dr. Fan in our clinical trials for the past 10 years. And clinical trials open up an option for owners that maybe wouldn't be able to afford cancer treatment otherwise. And a lot of times these pets get to stay with us for the week or for a few days. So you do get pretty bonded, and it puts the biggest smile on my face when they see me and they wag their tail.
We have a clinical trial that's being sponsored by a therapeutic company called Ancra Therapeutics. We have recruited and treated 12 dogs so far in this clinical trial. Many of these dogs have tolerated the therapy very well, so we feel that the therapy is safe. Excitingly, there are dogs that are showing great responses. So this technology is all about retaining a very, very potent inflammatory cytokine, interleukin-12, into a localized region, specifically the tumor or the lymph node that we're injecting. If you give interleukin-12 into the bloodstream, it is very toxic, and that actually halted its development for the treatment of people many decades ago. Now with new technologies, including Ancra Therapeutics technology, which is anchoring the interleukin-12 to an adjuvant called aluminum hydroxide, this allows us to retain that interleukin-12 in the local tumor microenvironment, have great effects at stimulating the immune system in the tumor microenvironment, but minimizing its escape into the bloodstream and therefore minimizing toxicity. The whole goal is to have a robust systemic immune response that not only fights the tumor locally, but has the capacity to actually shrink tumors that are far away from that primary tumor, such as lung mets or metastasis to the lymph node. The exciting thing is that the data that we've generated has been, I think, very important to continue to streamline and guide the future clinical design in people. And the technology has actually received IND approval on the human side. And the company is slated to start phase 1 human clinical trials with this technology at the end of the year. So hopefully we'll see some very, very interesting results. We hope that the studies that we did in dogs will be very important in recognizing what aspects we need to look for in people. And that would just further solidify the dog with certain types of tumor as a very good comparative tumor model to help guide the same therapies in people.